these truths to be self-evident.
That all men are created equal.
As a member of Congress, I get to have a lot of really interesting people in the office, experts on what they're talking about.
This is the podcast
for insights into the issues.
China, bioterrorism, Medicare for all, in-depth discussions.
Breaking it down into simple terms. We hold
these truths. We hold these truths.
With Dan Crenshaw.
The Eagle has landed.
Welcome back, everybody. We are returning to Hold These Truths, your favorite podcast, and you're going to give it 5 stars because it's your favorite podcast. And we've got a repeat offender here. We've got Dr. Scott Gottlieb. He's the former FDA Commissioner under President Trump, currently an AEI Fellow, member of National Academy of Medicine, served previous stints at the Department of Health and Human Services, was a professor at New York University School of Medicine. Since the last time we've spoken to him a couple times. The first time you may remember Dr. Gottlieb, he was all over the news during the year 2020, talking about COVID. He was one of those, I think, familiar faces that people look to for just get some balanced and takes on the information that everybody was receiving at a rapid fire rate. He's been working on a new book recently called The Miracle Century, Making Sense of Cell Therapy Revolution. This is something we take an interest in on the healthcare subcommittee on energy and commerce. So it's going to be nerdy, but I think there's going to be some interesting takeaways from what cell therapy is and how it can help our future generations. And help I mean a range of diseases from Alzheimer's to heart damage to cancer. Maybe even reverse the effects of aging, and then we can all be vampires. But all right, I'll stop there. There's a lot to say more about his bio, but Scott, thanks for being on.
Thanks a lot. Thanks for having me.
So, let's just start with the book. It's not out yet, but why cell therapies? You could write a book about a number of things, I'm sure. I don't know what I should know this, but I forgot. What did you specialize in as just in the medical field, and then did that relate to this book at all?
Yeah, general internal medicine, and I practiced as a hospitalist, taking care of patients inside the hospital, inside the intensive care unit. But coming out of the FDA, so I was at FDA from 2017 to 2019, and when I was there, we approved the first gene therapies. We approved 3 of them at that time. One was a treatment for a pediatric form of leukemia. We also approved gene therapy for a treatment for an inherited form of blindness. And what I was seeing with respect to not just what we were able to prove over that time period, but what was in the pipeline was the transformative potential of these new modalities of cell and gene therapy more broadly. And that's why I wanted to write a book focusing on the development of this technology. If you think about what we do in medicine and what I do as a hospitalist, a lot of what we do when we prescribe medications is treat the symptoms of disease. And sometimes we're fortunate enough to be able to slow the progression of a disease. There's very few places in clinical medicine where you can actually prescribe a therapy to cure a disease. Outside of oncology, certain forms of cancer that you can cure, there's curative therapy available for certain forms of cancer. And outside of infectious diseases, where you can treat an infection, for example, with antibiotics and cure the infection, most of what we do in medicine is prescribe drugs that treat the symptoms of disease, and in some cases, hopefully slow the progression of the disease. In surgery, you can potentially cure a disease, but not necessarily in medicine and internal medicine, the field that I practice in. And what cell and gene therapy offers, really for the first time on a broad basis, is the ability to reverse the effects of disease, and in some cases, develop a fundamental cure for disease. And we're already seeing that with some of the therapies that have been improved, and the pipeline is very full right now. So I think this is going to fundamentally transform medicine, and we're just at the early stages of it. And that's why I wanted to focus on it.
So, one kind of cell therapy that I think most people are familiar with is stem cell therapy, but I have no idea whether it works or not. I think there's a lot more clinical data that needs to happen, and it's unclear what it's useful for and what it isn't. Sometimes people take it out. There's different types of cell therapies or stem cell therapies. Sometimes they take it out of fat, kind of reproduce it. Maybe that's considered a more high quality one. And in other cases, they literally just you take it out of maybe your marrow or your fat and then just re-inject it into your joints. And then, maybe your joints feel better. It's kind of unclear. What's your general take on that? And then what else do we mean by cell therapies beyond just again, this sort of, I think, what would I call it? Yeah, this fad of stem cells.
Yeah, that was a pretty good overview you just laid out of the different modalities, if you will, of cell therapy. So you talked about taking cells out of the bone marrow. That's generally platelet-rich plasma, which a lot of orthopedic surgeons are using to inject into joints to try to rejuvenate joints. Then you talked about adipose stem cells. Those are being used also in orthopedic procedures and also some cosmetic surgery. The real breakthrough, I think, came in 2006 when we developed the technology to create what we call induced pluripotent stem cells. And that's basically the ability to take an adult cell, so a cell that you might derive from a person's own body, and
Right.
basically turn back the clock on it, turn it back into a stem cell. So take a finished adult cell and turn it back into its sort of primordial, original state, a stem cell that can then be induced to form other kinds of tissue. And so a lot of the treatments that are being put into development right now are based on induced pluripotent stem cells. So, those kinds of modalities, when you talk about stem cells and induced pluripotent stem cells and even adipose stem cells, a lot of that falls within the rubric, broadly speaking, of regenerative medicine, trying to use stem cells to restore tissue that may have become damaged or destroyed as a result of disease or injury. The other cell therapies that are in development, and that's what I focus on very heavily in the book, are the ability to take immune cells like T cells and re-engineer them and use them as targeted therapies, or to take blood cells and re-engineer them to help cure diseases like sickle cell disease or hemophilia. So that's a whole nother field where we're able to, by manipulating the DNA of blood cells, re-engineer blood cells either to treat diseases, or in some cases when you're dealing with a disease where the underlying dysfunction is in the blood cell itself, try to cure the disease. And we just saw a treatment get approved for sickle cell disease, which was a gene therapy delivered to blood cells. We saw one recently approved for hemophilia as well, another blood disease.
How on earth do you manipulate the cell's DNA? I mean, we talk about that in modern parlance, like it's something like, oh yeah, I mean, you just manipulate you just read the DNA. Nobody has any clue how to do that, though, of course. What are you injecting it with some kind of specific chemical or combination of proteins? Like, and where do you even get those proteins? I like to try to explain things to people just from the very basics, if it's even possible.
Yes. So one of the things that I did with this book was talk about the early. So I focused a lot on the development of CAR T therapy. This is a form of immunotherapy being used to treat different forms of cancer, particularly blood cancers, where scientists basically take T cells and, as you said, manipulate the DNA to get the T cell to express a new receptor that can recognize the cancer cells. And so you take these T cells out, you re-engineer them with new receptors, where you basically train them how to recognize the cancer, and then you expand them and re-inject them in large quantities. And when they get reinfused into the same patient that they were taken from, now with this new receptor that they're expressing, they go on to attack the blood cancer cells. The technology for trying to re-engineer and manipulate the DNA of T cells really started in probably the early 1980s with research coming out of Stanford and then MIT, where scientists figured out how to use viruses, and particularly retroviruses, basically like HIV. HIV is a retrovirus, to take to re-engineer the virus itself and take the harmful features out of the virus, but maintain the aptitude that viruses had, the capability that they had.
Okay, but that's exactly so. Like, how did they do? How did they re-engineer it? Or did they mix it? Like, did they mix it with some other chemical 1,000 times until they figured out, okay, when you do it this way, it actually re-engineers and has the effect that we want it to have? Is that somewhat accurate, or do I have no idea what I'm talking?
With the virus itself, they used laboratory techniques to splice out components of the virus's RNA. This was the RNA in the virus, because viruses express RNA, not DNA. And so take out certain genes and then reinsert other genes in the virus that gave it these new features, and then use the virus as sort of a Trojan horse to then inject that genetic material into human cells. And so what viruses do, if you think about HIV, what HIV does when it infects our cells is it injects its own genetic material into our cells, which then train our cells to make new copies of the virus. So they basically hijack our cells. And so if you can use that aptitude of the virus, the ability to hijack a cell and
Mhm.
inject new
So you
genetic
only need
material.
one. But you kind of make it sound like you're performing surgery on a single cell. Is that
I mean, that's a good way to think about it.
and they're kind of literally like poking out the RNA they don't like and somehow putting in the RNA they do like, and it's just, I mean, like I don't want to dig too much deeper into this. I'm just curious. But it's just amazing what we've been able to do I don't even know if our TVs turn on. So I mean, this is no, we don't know how much of anything gets done. But it's fascinating. So okay, so those are another couple different types of cell therapies, and I think we crossed over in that conversation into what you would call gene therapies then also, right? Because you were just talking about
Yeah,
splicing out RNAs and DNAs.
when you think about cell therapies, a lot of the cell therapies that I'm focusing on, a lot of the cell therapies that you see being approved, where what you're doing is manipulating the genes in a blood cell. So you're extracting a blood cell from a person or an immune cell, a B cell or a T cell, manipulating the DNA of the cell to give it new features, and then reinserting that same cell into the patient. That's really a form of gene therapy because the gene modification you're making isn't directly on the patient, it's on their blood cells, and so you're extracting their blood cells, modifying them, and then reinserting them. There's other forms of gene therapy where what you're delivering to the patient isn't necessarily a modified cell. What you're delivering is the vector itself. So you're delivering the virus directly to the patient into some tissue, some organ in the patient, and then hoping that that virus will then deliver the new genetic material into the right cells. And so when you think about, I talked about a gene therapy that treats an inherited form of blindness. In that case, what you're doing is literally injecting the virus particles that have been modified to insert this new gene directly into the patient's eye. So some of these gene therapies, you might insert the virus directly into a patient's liver or their pancreas if you wanted to try to treat diabetes or try to treat certain diseases that manifest themselves in the liver tissue. So those are two different forms of gene therapy. I think of the manipulation of blood cells like CAR T therapy, which is what I write a lot about, is sort of an exogenous form of gene therapy. You're doing the gene manipulation outside the body in a person's blood cells in a laboratory setting. And then endogenous forms of gene therapy, what you're delivering is the viral vector to deliver the gene modification directly into the patient's tissue.
And for those T cell therapies, is that related to the cancer treatments mostly that I get, or just
A
any
lot of the
immune
CAR
disease?
T, yeah. So both. Originally, the CAR T therapies were approved for the treatment of different forms of blood cancer. Now, what we're finding is some of these CAR T therapies that are effective at treating blood cancers are also potentially effective at treating autoimmune diseases. So there's some trials going on looking at CAR T therapy in, for example, lupus, where you're seeing some really profound early results. None of them are approved yet for those indications, but there's really encouraging clinical data. And the belief is what the CAR T therapies do is really, so this CAR T therapy in particular, what it does is wipe out a patient's B cells. And so if a patient has a cancer that arises in the B cells by delivering this CAR T therapy, which targets an antigen found on the surface of B cells called CD19, you're able to basically ablate or wipe out all of a person's B cells, both their healthy ones as well as the cancerous ones. And that's how you cure the cancer. But what we've also found is by wiping out the B cells using these CAR T therapies, what we think happens is you reset a person's immune system. And
Mm.
so for certain patients who have autoimmune diseases, you can effectively extinguish the autoimmune disease. And we've seen that in very small, albeit early clinical trials, but very profound results in a group of patients.
So would it be safe to say that if there is a cure for cancer on the horizon, it's probably going to be in the form of some kind of gene therapy? Is that an assumption that you might make at this point, or at least some cancers?
Look, we've seen because of all the progress that's been underway for many years, we've seen a lot of what I would call cures for cancer being developed and approved by the agency, including CAR T therapies. In some patients, so the particular CAR T therapy that was approved when I first joined FDA in 2017
Hmm.
cured certain patients who had acute lymphoblastic leukemia, which is a form of cancer that affects blood cancer that affects a lot of young patients. It's a pediatric form of blood cancer typically. Some patients who got the therapy didn't benefit from it, but there were a number of patients and continue to be a number of patients who fail other therapies that are available, fail conventional chemotherapy, and can be cured by this CAR T therapy. So I think we're going to see that more and more, where these cell therapies are so effective and so precise in their ability to target cancer that you're going to see more cancers able to be cured. And really the holy grail right now, we've been good at designing these cell therapies to treat blood cancers. What we haven't been able to do yet in a really reliable fashion is use these cell therapies to also target solid tumors. And you're seeing now better methods for doing that. It's going to take some regulatory innovation on a part of FDA, but also take some scientific innovation because the platforms for doing that are different than what we've done in the past. But you're seeing some encouraging results in clinical trials where these CAR T therapies are now being trained on solid tumors. There was some data out about 12 months ago looking at a CAR T therapy in the treatment of pancreatic cancer, which, as you know, is a typically deadly form of cancer.
So I mean, if you sum this up in layman's terms, you're always, correct me if I'm wrong, in every single one of these cases, you're using the patient's own cells, and then you're putting them through some kind of modification, the kind of modifications that you've already described. I'm not going to try and repeat them, and then you re-inject that into the body, and so it's a very personalized form of treatment in that sense. Is that always the
That's
case,
right.
or do you
And
sometimes use other cells from somebody else?
yeah, you can use other cells, and there's treatments in development that are sort of off the shelf forms of cells. And the virtue there is that if you can develop an off the shelf preparation of cells, it's something that you could manufacture on a large scale and stockpile, store in a
Yeah.
pharmacy, rather than having to personalize this treatment, as you said, for every individual patient, take the patient's own cells out, manipulate them, expand them, then reinfuse them. You can understand how difficult and costly of a manufacturing process that is, and it adds to the high cost of these therapies. The advantage of using someone's own cells, though, is that they're not going to be recognized as foreign.
I do think we're taking a step back sometimes looking at these therapies and thinking like all of these steps and research and innovation and breakthroughs that had to happen to get to this point. I mean, to even recognize that some diseases are related to your genetic makeup. I mean, so the, going back to the Human Genome Project and the necessity to actually map out everyone's genetics and then understand how to, do surgery on DNA, which I still really don't I can't conceptualize what you're saying. And I don't know. I mean, like with a scalp, like with a really tiny scalpel. I mean, what are you even talking about? Or, I mean, there's just so many steps. And I think it's always good for people to take a step back and show a little gratitude for the kind of modern lives that we live, where people have taken these little baby steps over the course of hundreds of years to get to this point where you can literally edit genes and save you from a disease that would have killed you just like that. And you would have no idea why, even just a few decades ago. That's kind of interesting.
Look, I think that's the key point here, and that's the point I tried to make in the book by looking back over this history. If you look at the history of the development of the first CAR T therapy, so I mentioned that the first CAR T therapy was approved in 2017 for the treatment of this pediatric form of leukemia. If you look at the drug was called Kymriah. If you look at the history that led to the development of that drug, it really stretched back 30 years. The first recognition that T cells could be effective in targeting cancer was made probably in the 1970s, through research that came out of studies with bone marrow transplants, where scientists realized that when patients with cancer received bone marrow transplants, in cases where the bone marrow wasn't a perfect match for that patient, those patients actually were more likely to be cured of their cancer. And so what they discovered was that there was a therapeutic effect from the marrow itself. There was something called graft versus tumor effect. So the marrow was attacking the tumor, and so was stimulating an immune response against the cancer that the patient had. And then they isolated the cells in the marrow that was responsible for that immune attack, and it turned out to be the T cells. And so then scientists realized, okay, T cells could be effective at targeting cancer cells. How could we harness this ability? And that's where the idea came for trying to take T cells and re-engineer them to train them how to target specific kinds of cancer. So that journey that I just described was literally a 30-year journey where there were these small incremental insights that scientists were making along the way, and in new capabilities that they were acquiring, like the ability to use retroviruses to deliver genes, or figuring out how to manipulate genes themselves in cells, that ultimately led to the first approved therapies.
Yeah, and you know that story applies to most modern amenities that we have, whether it's in healthcare or this computer screen that we're using to chat with each other, speed of light. So we'll talk about regulation for a little bit. This is a little wonky, but it does serve right into what I do up here. We're always trying to figure out how we can be more innovative, or I guess more conducive to innovation at the FDA, and my committee oversees the FDA. Um, but I don't. It's hard for us to find a lot of easy buttons to say, like, let's just do things right and awesome. That's hard to find, at least from the legislative perspective. You laid out two different scenarios earlier, which I thought would be a good starting point for this conversation. Which was, in one case, you're taking my own cells, you're manipulating them a little bit. And was a question of what is a little bit and what is a lot of bit. That's important for how we talk about this. And then in another case, you're taking just general cells, and you're creating a drug that you can put on a shelf, as you mentioned before. It's just more efficient that way. But those are two very different things. In one case, again, it seems to me like you're talking about like a bone marrow well, is that a bone marrow transplant, or even just is that, I guess I don't know enough about bone marrow transplants to make that analogy, but you're talking about my own cells, somewhat manipulated back into my body. In another case, you're talking about
Yeah.
somebody else's cells. So it sounds a lot more like a drug, whether
Yeah,
that's
the
a
bone marrow
biologic.
transplant is a good analogy for
Okay.
the first scenario, where it's your own cells.
Right.
Yeah.
Right. Because it is your own cells. I'm not crazy. Okay.
Right.
Haven't done one before, so I found myself talking a bit out of turn. That's probably a good thing. It means I've been a patient a lot, and thankfully not for that. Okay, so now there's a real question at FDA over how that should be regulated, and because you've got all this really neat innovation, and um, for it can take, I don't know, you tell me, like, how long is it taking for some of these gene and cell therapies that could potentially save lives, change the world? How long is it taking to get them approved? Do we have the right way to approve them, or are we thinking about them too much like a drug? I dealt with this because in one of my own constituents does this the stem cell therapy that we were talking about, and they take it from your fat, and they spin it around a centrifuge, extract the stem cells, and then get the stem cells to kind of grow on their own. So you reproduce your own stem cells, then you can re-inject those at a later date. It doesn't, there's not a whole lot of other manipulation there going on, and yet it is treated, as I understand it, like a whole new drug application. And then, just from
Yeah.
my non-doctor, common sense standpoint, that seems a little out of touch. But what's your take on that?
Well, look, you touched on the right principle. So if you're talking about the off-the-shelf cell preparations, in most cases, those are going to be. I can't think of a scenario where it's sort of an off-the-shelf preparation of cells that have been engineered aren't going to be a drug. But when you're talking about cells where you're extracting the cells from the patient, doing some form of manipulation to the cells, and then reinfusing them into the same patient, and potentially in the same spot where you extracted the cells, it's the question of when that crosses over into becoming a drug isn't always clear to everyone. The way it's defined in regulation is if the cell that's extracted from the patient is more than what we call minimally manipulated, then it becomes a drug. And so the question is, what is more than minimally manipulated means? The way I think about it and the way I describe it is, if you're changing the characteristics of that cell, if you're doing something to the cell where you're changing the behavior of the cell so that it behaves in a new way once it's reinserted into the patient, that is a general way to think about what is more than minimal manipulation. So it might be changing the physical characteristics of the cell. It might be adding drugs to the cell to change how it behaves. It might be changing the genes of the cell. That's certainly more than minimal manipulation. But there are these things that fall close to the line. And I think that's where a lot of the debate is. And it sounds like you might be describing one of those circumstances with your constituent. So, for example, if you extract adipose stem cells and you might reinsert them to try to treat an orthopedic injury or to have a cosmetic effect, if you're taking the cells, extracting the cells from the patient and spinning them down so you concentrate them and then reinfusing that preparation, that might be more than minimal manipulation in the sense that you're changing the characteristics of the cell because you're taking it out of its original scaffolding, and that will change the characteristics of the cell, but not in a very significant way. And so that might be one of these sort of on the line kinds of questions about whether or not it crosses a line and becomes more than minimally manipulated. And these are kind of the gray area questions where I think sponsors, have to come into the FDA, ask for permission. And in certain cases, it could be frustrating for them because they don't feel like they're getting back a timely answer. When I was at FDA, what we tried to do was carve out certain of these, what we considered low risk circumstances. So, exactly what I described, a situation where someone may be taking adipose stem cells, concentrating them. So it's probably more than minimal manipulation because they're changing certain characteristics of the cell by taking it out of its usual scaffolding, but using it in a low risk way, like a dermal procedure or like an orthopedic procedure. So we explicitly carved that out and we said, look, this may be more than minimal manipulation, but for purposes of regulation, we consider it a low risk use of cells. As long as you follow good manufacturing standards, you do it under sterile technique, there's not a lot of risk to the patient. So we're going to choose not to regulate that. We're exercising enforcement discretion. And I think that's where the agency can lean forward a little bit more and describe more of these sort of low risk circumstances where it chooses not to regulate, so that it could focus its activity on the higher risk circumstances where there's more potential risk to patients that are being introduced by some of these therapies.
Yeah, I mean, it gets to this uncertainty of on the regulatory side, and I think a lot of the private sector feels like there's not enough consistency to how these decisions are made, whether that discretion is given or whether it's not, or whether they place them under a different pathway that is just far more cumbersome and therefore far more expensive. And I've seen other cases too where it seemed kind of obvious that the reviewer in charge just wasn't the right person to assess that particular technology. Like they just didn't have the background. And so what do they default to? They default to no, and to no again, and to another trial here and another trial there. And next thing you know, you're in another round of fundraising for your company, which is about to go broke. And then we'll never have that technology again. So that balance is extremely important.
The biggest challenge that I saw when I was at the agency, and I think it persists, there's certainly challenges on the clinical portion of the review where you're trying to demonstrate that a product is safe and effective for its intended use. And so you're running clinical trials to try to generate clinical data, and there's always questions about what is the bar, how much of a therapeutic benefit do you need to demonstrate, how many patients is that going to require, especially when you're dealing with these very expensive modalities like cell therapies. Those trials can get hugely expensive. Putting a single patient in a clinical trial for CAR T therapy can literally cost $1 million. And so, if you're enrolling 100 patients or asked to enroll 1,000 patients, that's a pretty big delta if it's $1 million for every patient that gets enrolled. But the bigger sort of bucket of uncertainty, in my view, was around the manufacturing issues, where I think it was very hard for the FDA, and still is hard for the FDA, to recruit and retain people who have expertise in how these products are manufactured and understanding all the complexity involved in that, and also understanding how small changes in manufacturing, where you might be trying to change your manufacturing process to get a better yield of these cells or be able to scale it up more quickly or make it less expensive, how all those little changes is going to translate potentially into changes in how the cells perform. And so because the agency doesn't have a lot of expertise in that, and there isn't a lot of expertise in that because this is such a new science, what ends up happening is when these sponsors want to make slight modifications in how they manufacture these cells, which is very important to trying to scale it up in a way that you can actually turn it into a therapy that could be commercialized. And in many cases, FDA is going back and asking them to reproduce some of the clinical data to be able to demonstrate that the characteristics of the cells haven't changed in a way that's changing their safety and their their efficacy. And that's where I think a lot of sponsors are getting stuck, and that's where a lot of the costs are being added. And that's where we need to focus more on more regulatory policymaking to make that process itself, more efficient. And this is the problem with a lot of early technologies. We have a brand new platform. I remember back in the early 2000s when antibody drugs were coming onto the market, they encountered some of these same complexities where the FDA really didn't understand how changes in how those antibody drugs were being made translated into changes in how they performed clinically. And so asked sponsors to go back, asked companies to go back and do a lot of new clinical trials every time they made a change in the manufacturing. And it was an impediment to getting a lot of those products scaled up and on the market as well. Now we have the tools to do that. And so you don't need to replicate clinical trials to prove that one antibody is exactly the same as another one, even though the manufacturing process might have changed. And we feel so certain about our ability to determine that now we're approving generic forms of antibody drugs without requiring large new clinical trials every time.
Just out of curiosity, what's that technology that changed? Just is it laboratory technology? Your ability to
Laboratory
just
technology, yeah.
okay,
So it's a lot of applied technology. So things like mass spec, a tool for looking at protein drugs, looking at antibodies, and being able to determine just based on the physical characteristics that this antibody is going to perform exactly the same as that antibody. So
right?
now we're able to impute by looking at the structure of the antibody how it's going to behave. So it leads to a policy point though, because you said there isn't sort of a magic bullet. And many times policymaking in this realm isn't satisfying in the sense that there's not one thing you can change and that's suddenly going to unlock the whole system. It's a lot of small modifications to the regulatory process that's made over years by Congress that,
Yeah.
gets accrued and eventually consolidates into new ways of doing things. But I think where we can make a big dent into some of these uncertainties that we're talking about is investing more in that applied science for answering these regulatory questions. And that's not something NIH does typically. It's not something that companies do because they can't own the intellectual property around that. There's not a profit to be had in that. And so it's really this sort of barren landscape, if you will, where no one's really focused on the applied technologies to do things like be able to demonstrate that one cell is exactly the same as the other for regulatory decision-making purposes. And this is someplace where I think NIH could be focusing more of its resources. NIH is looking at getting more into drug development. They want to be engaged in some of the
Yeah.
sexier aspects of the sort of scientific enterprise. But this applied science is really a gap where I think they could be focusing more attention.
That's interesting. I mean, they have plenty of money. I don't think they need more money. I think they get more money every year. And they have a lot of grant programs. You would think that they could invest a little bit on that. And by applied sciences, again, so what we mean is the kind of technology that allows you to sort of replace a clinical trial, which are
That's right.
extremely expensive.
Exactly. A
So
new way to measure clinical benefit using a laboratory tool, or think of like Alzheimer's disease. If we could figure out how to use MRI and functional imaging to assess patients and diagnose when they have Alzheimer's and how it's progressing, rather than requiring clinical trials where you're measuring a patient's physical performance on mini mental status tests, which takes a long time. And by the time you see changes in that, there has been a lot of disability that's accrued to that patient. So if you can measure, decline in function
Mm.
more quickly using imaging, that would help accelerate clinical development of treatments for Alzheimer's. But we haven't validated those tools in a way that regulators would rely on it. That's something that, NIH does do some of this. So I don't want to say they're not doing it, but I think it's someplace where we could be focusing a lot more resources on validating some of these laboratory tools.
Speaking of prices, I mean, you mentioned $1 million per patient for some doses. So, I mean, that's hard enough in a clinical trial. So, but that also makes me assume that if that were approved as a therapy, that's still $1 million per dose. But is it a one-time dose that we're talking about, or is it? I mean, these things are really expensive, and I guess
Yeah.
my first question is why? Is it just the technology and amount of labor and personnel that's required to do it, and then how on earth do we pay for this once we find these breakthroughs?
Yeah, so the $1 million per patient that I was referring to was the cost of a single patient in a clinical trial with something like a CAR T therapy. But you're right, some of these gene therapies that have been approved have been priced at more than $1 million. Have been multi-million-dollar gene therapies. Typically, some of these are administered as a single dose or maybe just a few doses. Um, the challenge is going to be paying for these, especially in austere uh, insurance schemes. And so what we've seen is very differential access in the marketplace where patients who have better insurance, private market insurance, have had more timely access to some of these therapies when they've been approved, and people who are in more austere insurance schemes, and particularly Medicaid, have not had timely access. Those insurance schemes have been much slower getting coverage in place. And when you're dealing with a gene therapy or a cell therapy like a CAR T therapy that is literally curative or potentially curative for patients, or helps arrest a disease like Duchenne's muscular dystrophy, where patients accrue disability very rapidly, young patients who are born with these inherited diseases accrue disability very rapidly. The timeliness of access can literally be the difference between life and death, or a life lived with disability and one lived, less
Yeah.
encumbered by disease. And so, this is going to be, I think, a big challenge for us. Why do they cost so much? Part of the reason is that they just cost a lot to make. So the cost of goods is very high. If you think of a small molecule, a pill drug, it might cost pennies to make that drug, or maybe dollars to make that drug. And if you're charging $100,000 for the drug or $50,000 for the drug, a lot of that's profit because the drug didn't cost much to manufacture. With some of these cell therapies, literally the manufacturing process can be tens, if not hundreds of thousands of dollars. So the cost of goods is very high. And what we've seen in this space, and you have seen venture capital sort of pull away from these areas, is because the returns haven't been that good. If you're looking at sort of relative investments in different kinds of drug modalities and different kinds of opportunities, while these therapies are hugely exciting from a clinical standpoint and have the ability to cure disease, they're typically targeting very small diseases, some cases rare
Right.
diseases. And so you're not going to be prescribing them to a lot of patients. The cost of developing and manufacturing them is very high. And so companies that have invested in these spaces have not had good returns, and a lot of them have curtailed some of their programs. And so that's been one of the unfortunate things. I think it's changing now because the opportunity set's getting larger. But if you look back over the last maybe 5 years, a lot of the big drug companies and the venture capital firms that were investing in cell and gene therapies pulled back for a period of time.
Have you seen any effects from the Inflation Reduction Act legislation? Within that legislation was the requirement for Medicare to negotiate with new therapies, new drugs. There's some exceptions in there. I don't know if these cell and gene therapies would fall into those exceptions, but it's a requirement to negotiate at the price that Medicare wants to set for you, and there's a pretty large punishment if you don't take that negotiation, being a 95% tax on your revenue. So have you seen that affect the market and the venture capital especially in the investment space?
Yeah, not directly with the cell and gene therapies. I mean, indirectly, insofar as there has been a reduction in R&D budgets across the industry as a result of the IRA. But where I've seen that the IRA have a more direct impact is biasing investment away from small molecules, because the negotiation gets implemented sooner for small molecules versus biologicals like antibody drugs or even these cell therapies. You are seeing companies pull away from investment in small molecule pill form drugs, particularly small molecules that treat drugs where it's going to be heavily reimbursed by Medicare. So drugs, for the treatment of conditions that would affect older individuals.
Mm.
There's definitely been a reallocation of capital, and you see that in the deals that are getting done in the pharmaceutical industry. So the thing that's going to get affected first is the deal making, where a company's placing new investments, and they're now shifting investment towards large molecules, antibody-based drugs, because those come under the price negotiation later than the small molecule drugs. And so they're sort of free from the price controls for a longer period of time. And that's unfortunate because there's certain diseases and certain targets, biological targets, that can only be addressed with small molecules. If you're thinking about trying to target something inside a cell, so an intracellular target, if you're thinking about trying to target something in the brain that has to cross the blood-brain barrier, small molecules are a very efficient way to do that. When we're talking about large molecules, biologicals like antibody drugs, typically they only target things on the surface of a cell. They can't get inside the cell. And so there's whole diseases and whole areas of biological targets that can only be addressed with small molecules. And that's a real concern. I think a lot of us have a concern that now there's going to be less investment in those areas, and we're already seeing it.
Um, so can the small molecules always mean pill form, or can that also mean cell and gene therapies? We're just talking
Small
about size.
molecule, I mean, yeah, the cell and gene therapies are biologics, and so they would be considered large molecules. Small molecules are generally pill form drugs, but some of them are also infused. There's small molecule drugs that are infused or injected. The GLP-1s, the drugs that are being used for weight reduction, are peptides. So they're an injected drug, but they're considered a small molecule drug. So they're subject to the IRA price negotiation in that shorter time frame. In fact, Wegovy, one of the drugs, semaglutide, is going to be subject to the price negotiation next year. So small molecules can be injected drugs, but they're typically swallowed as pills.
I wonder too, and again, there's no way to know this because we don't have access to the multiverse, and we can't see what history would have been without some particular law being passed. But because there's still a timeline on when they're subject to that negotiation, do these companies? I mean, it's hard to know. I don't think they would announce it, but they clearly have an incentive to have a higher price from the get go if they know they're going to get hammered by CMS at a later date. Are you seeing that at all?
The market's competitive, so it's hard. There's a lot of downward pressure on pricing right now, and if you look at inflation in the drug sector, last year was the first year that prices overall fell, and I don't attribute that to the IRA. I just attribute it to the competitive pressures in the marketplace exerted by PBMs and insurers, and so it's hard to say what would have been. I think where it's easier to see an effect. I mean, you could certainly there's certainly an argument to be made that as we introduce more price controls in the marketplace, and the IRA is certainly a price control, it creates incentives to try to launch at higher list prices because your price is going to be driven down by the various price controls that are in the marketplace. You're going to have a shorter window to recoup your
That's
investment.
what I mean. Yeah.
Where I see the most immediate impact, though, is just the flows of capital and what companies are choosing to invest in. And companies aren't abandoning programs that they had, so you're not going to just shut down a program that's in later stages of development because of the IRA.
Right.
But you're not going to be putting new capital in those areas. And once companies get out of the scientific know-how of developing small molecule drugs, so once they get rid of what you call combinatorial tree, they're not going to just get back into it. They're going to lose the expertise and the know-how for doing that.
Well, what diseases should we be most worried about in this space? Because I'm not despite all my knowledge on this, I don't know which diseases are more likely to be treated with small molecule drugs. So, what is that? What might not get that investment?
And so, one area that I've flagged is mental health diseases, where a lot of the targets are intracellular targets, and so can only be addressed with small molecules. Infectious diseases, things that we have to treat inside the brain.
Or we're talking about like Alzheimer's.
If you're trying to address it with a small molecule, I mean, some of the drugs that are coming onto the market are biologicals that are being used to target brain diseases like Alzheimer's as well, neurodegenerative diseases, but a lot of them are small molecules. So anything like that, anytime you're trying to get inside a cell to target some receptor inside the cell. And so, it could be cardiovascular drugs as well, and certain drugs just used in primary care. But the mental health diseases is one big area that I worry about because a lot of those are intracellular targets that would typically be addressed with small molecules.
You've worked a lot with your European counterparts. I don't know what the FDA is called in Europe, but there's pros and cons to it. And I just got back from London and on an intelligence committee trip, so it nothing to do with healthcare. But their politics are all riled up as they are in every country all the time, just like they are here. And one of the biggest issues there is NHS, their National Health Service. And, which is apparently in disarray, falling apart. And there's months and months and months of wait times for whatever it is you need. I don't know, maybe, I don't know if there's a question in there, but maybe I'm looking for a comment and what are the pros and cons of our system Because I do know that sometimes it's easier for innovators to go to Europe. I've heard from the private sector that sometimes they just have an easier time dealing with European regulators in some instances. Uh, at the same time, I think America's by far number one when it comes to innovation on anything with respect to healthcare. So talk to me about that. What should we be wary of when we look at, the continuing downfall of NHS, and also what you see in Canada with what is effectively a single payer system, not quite like NHS. But give us your thoughts on that if you don't mind.
Yeah, look, I think that the European, the EMA, the European regulators for a number of years were leaning forward a little bit more on the approval of treatments for significant unmet medical diseases, particularly orphan diseases, inherited diseases that affected pediatric populations. I think so. There was a gap between FDA and EMA in terms of, how permissive they were willing to be when it came to the approval of novel therapies that treated very serious unmet diseases where there weren't good available therapy. I think a lot of that gap's been closed. I think 21st Century Cures and the implementation of it has closed a lot of that gap, and that FDA has adopted some of the same approaches that the Europeans have. So I don't think that the European regulators, I would argue that they're more flexible now. I mean, there may be certain pockets of certain therapeutic categories where they might have a different focus than the FDA and perhaps are a little bit more accommodating. But I think overall, um, at worst, the regulatory authorities are comparable. And I would argue, and I worked at FDA, so I'm going to defend the agency to some degree. But I would argue, I think FDA is a more accommodating agency overall. With respect to Europe, though, these new products just aren't getting paid for. And in more and more cases, you're seeing companies just not launch them in Europe or launch them much later in Europe. And, particularly when you're dealing with products that are this potentially transformative in terms of the therapeutic effect that they're delivering, that's going to start to translate itself into differences in outcomes. And you saw for years, and I wrote about this a number of years ago, you saw a real divide in cancer outcomes between the US and UK. And
Hmm.
while it's hard to prescribe exactly what that's attributable to, a lot of some of it's early diagnosis and differences in diets and environment. But certainly some of it was attributed to the fact that there was this whole spate of drug approvals in 2000s and sort of the mid 2000s that were really transformative, and they weren't approved as quickly in the UK as they were in the US. Herceptin for breast cancer, Erbitux for colon cancer, Avastin for colon cancer, drugs that really had a big impact once they reached the market were delayed coming onto the market in the UK, and that divide has gotten wider over time, and I think you're going to continue to see that play out in terms of outcomes.
Yeah, and this gets, of course, to the debate of single payer systems versus non-single payer systems, and the constant reminder that, well, Americans are going to Canada and going to Mexico to buy their drugs, and I think it's important to say, the other side of that, though, is that the Canadians and the British are coming to MD Anderson in Houston when they have a really, really rare, difficult cancer. They're not staying in their home country. They're coming here because we might be buying some kind of generic, well-known drug there because their system is different, it's just easier to buy it. But for quality healthcare, they have to come here. And so which side would you rather? I'd rather be in our situation than their situation. And I think trying to find that healthy balance between the two, especially making, I think, old technology more affordable. And that gets to a discussion about, generic drug approvals and whether those are therapies or medical devices, whatever it is, and making that a more competitive system. But you also can't kill the goose that laid the golden egg and kill the innovation that gives you the healthcare in the first place. You can say that
Look,
healthcare is free. Go ahead.
I think there's a lot we could be doing to make older drugs more affordable, particularly after patents have lapsed and drugs should be subject to generic competition. In many cases, they are, but in some cases they aren't, where Congress intended for there to be brisk competition from generic alternatives.
Yeah.
You're not seeing that enter the market, and there's some policy and regulatory reasons why in certain cases that's not happening. But when you look at what's happening in Europe with respect to some of these, more expensive, but more advanced therapies, the ones we're talking about here, cell and gene therapies, but also a lot of the new biologics that are coming onto the market, patients just aren't getting access to it. And, for a long time, and I followed this pretty closely, for a long time, there was a real, revolt's the wrong word, but there was a recognition among patients in Europe that they weren't getting timely access to some of these new therapies, and you saw them pressuring their governments. That's died down. You see less organized pressure from patient groups in Europe who aren't getting timely access to some of these new therapies, because I think there's just been this sort of recognition that has set in that they just can't afford them. And companies in many cases just aren't launching the drugs in those countries as quickly as they'll launch them in the US or, Japan as well.
It's sad because you could also, so if a drug is, if a therapy, if this cell therapy is $1 million, and you're telling me that's largely because of the actual, I guess, laboratory process that is required to even do it.
Some of it,
It's an expensive, but of course, a lot of it is paying back their investment, which was
Profit,
sometimes in the billions. I mean, especially for the kind of clinical trials we were describing earlier. That the overall price per patient could be lowered if the market was bigger, right? I think that's the general economics of it. But we're stuck in this position where they either way undersell it to European countries, or underprice it to European countries just to get it out there, which sounds like it can't even be done with cell therapies because it's not like a pill. It's the actual process of doing it's very expensive. And so you can't scale. I guess the point I'm making is when you can't scale, you can't lower price. And so we're stuck, but hey, at least at least we have something. And, figuring out how to pay for it, is a whole other issue. I have it in my notes here. I don't know if you wrote about this or my staff just wanted to ask you about it. But on the warranty, is that an innovative way to think about paying for some of these extremely, expensive therapies?
Yeah, I think especially when you're dealing with a gene therapy, where it's sort of in many cases, it's going to be a one-time administration and you're hoping to cure a disease. But if you think about it, you're not going to be delivering the therapy over the course of 10 years or the lifetime. So you have to recoup all the investment, all the returns in that single administration. And that's why the cost of these therapies are so high. In those cases, what you might want to do is try to amortize that cost out over time. And so it's something like a warranty where, if the treatment isn't durable, if the gene therapy, if the effect that it's intended to have doesn't last 3 years or 5 years or 10 years, it doesn't offer a durable cure for the disease like was anticipated at the outset, the company might owe back some of the money. So rather than load it all into that single administration, the company's still on the hook for the performance of that product over time. Because in many of these clinical trials, when these gene therapies are coming onto the market, part of the uncertainty is you don't know whether or not the effect's going to be durable. The clinical trial might last 2 years and the patient's clinical response might last 2 years in the clinical trial, but then 5 years out, is that effect still going to be there? And in fact, we've seen with some of these therapies that the benefit wanes over time. And so a warranty or finding a way to amortize the cost over time is one way to deal with those very high upfront costs that aligns with some of the uncertainty around benefits that the products intend to deliver.
What would that do? I mean, it wouldn't make the upfront cost different, would it? Or how does it change the incentives to improve the situation? Maybe I'm not following it.
Yeah, well, in its simplest form, you might think of something where the company gets paid a certain amount over 5 years, and let's say the insurance company will pay X amount in year 1, X amount in year 2, out to 5 or 6 years. And they'll say, well, if the benefit starts to decline in year 2 or year 3 or year 4, then we won't owe those other payments. So that way, instead of paying all the money up front, they spread it out over time, and the company was on the hook for the performance of the drug over that period of time.
Oh, I see.
That would be the simplest way, yeah.
Right.
There's other kinds of financing schemes that you could think about, but that would be the simplest. But it becomes cumbersome because what the insurance company will say is, it's going to be hard to follow the patient for 5 years. We're going to lose that patient. They're going to switch insurance plans. How do we continue to follow them? So then you have to think of how do you create intermediaries in the market where you can continue to collect that data and hold the drug company sort of accountable for the results. So
Yeah.
they're harder to implement these things as a practical matter. They make a lot of sense as a policy matter.
Well, that's the whole thing about value-based medicine, right?
Yeah.
As opposed to fee for service. It in theory sounds great. In practice, that is very difficult to implement because value is inherently a word that is subjective in nature. To place value on something is usually there's usually some kind of subjectivity involved. And I think that's why the system often defaults to this sort of fee for service coding where, okay, I do this procedure, I get this much money, I know that ahead of time, et cetera, et cetera.
Focusing
There's
on the inputs rather than the outcomes. Yeah, because it's easier to measure the inputs rather than the outcomes.
very much so. And I don't know, I don't know if we figured out how to do that kind of value-based care, or do you think we have, or some insurance companies well ahead on that? Is there a good model that we would look to? I know we're changing subjects pretty dramatically here, but
I think Medicare and other private insurers have started to do it more effectively, and particularly when you look at the delivery system now, where more providers are capitated, meaning they're getting paid a lump sum and taking risk. They're starting to invest in ways to measure their clinical outcomes and their clinical performance over large populations. And so I think that the tools for measuring the things that you would tie the payment to have gotten a lot better. And you're starting to see more insurance contracts get written based on those outcomes, the measures of those outcomes.
what about last question before we go? It's been an hour. How do you think AI starts to play into this entire conversation, especially with FDA regulation? We've had a few hearings on it, a few discussions on it in the committee. My problem with the discussion of AI is always that it's oftentimes that we're just talking about advanced software and not really AI. I think the real interesting part about how AI could play into healthcare is when we start talking about diagnosis. And
Right.
we're going to get to a point where you have a conversation with an AI doctor and they diagnose you. And will that ever be an FDA approved decision? Who knows? And how do you even? I guess there's ways to test it, of course, but I mean, what do you think on it?
Yeah, look, I think it will be, and I think it can be. So, separating out AI on the drug development side, the drugs that come out of some of these natural language processing, these large language models, I think there will be drugs that are where an AI augmented process are helping to develop drugs. Those drugs are going to be put through a traditional regulatory scheme. They're still going to have to go through clinical trials. And if anything, I think the AI tool in that scenario is going to help explain the biological plausibility of the drug, why the drug should work in a certain disease,
Mm,
because what AI tools solve for is plausibility. And so to the extent that there's a better understanding of why the drug would work in that
mm,
certain disease state, that actually could be helpful in making the regulatory process more streamlined, because the more that FDA understands about why a drug should be working in a particular circumstance, and you go on and demonstrate that with clinical data, that gives the agency more confidence. On the medical device side, so using an AI tool, a large language model, to actually interact with a patient and help facilitate diagnosis, in many cases, even potentially make the diagnosis. I think the technology is sufficient right now, probably to be using these tools in certain lower risk, more routine circumstances. The challenge is nobody really wants to take an LLM through the regulatory process because people are unclear about how FDA would approach it. And so they're positioning these large language models right now as what they call clinical decision support software. So they're saying, well, this AI model is to help inform the provider and give the provider more insights. And so you might go onto a telehealth platform and your initial interaction might be with an AI tool that's asking you questions and formulating a presumptive diagnosis. And then it now passes that information on to the doctor, and then the doctor interacts with you or the provider interacts with you. So the tool in that circumstance is for clinical decision support. There is the capability, I believe, in certain lower risk settings for the AI tool to not only interact with the patient, but even prescribe therapy. If you think about what the chief complaints are that most people go to a primary care physician with, it's pain on urination, back pain, belly aches. It's routine things. Some of them could be serious, but the AI tools probably are sophisticated enough to know when to default to a provider and when it's something that's more routine based on the questions that they
Yeah.
ask, where it can be dealt with more in a more routine fashion. Um, how do you get regulatory approval for that? FDA is not going to be able to rip apart the AI model. It's black box, and they're not going to be able to figure out how it's working. And so the regulation needs to be put on the front end and the back end. What I mean by that is FDA needs to figure out, how do we regulate the training sets, the clinical data training sets that these AI tools are trained on, to make sure that the data itself is reliable enough, is representative enough, so the AI tool is being trained on an appropriate body of data. And then how do we query the AI tool with a sort of standard battery of questions to make sure that the answers that it's giving are consistently accurate, or accurate enough, as accurate as a provider would be. And that regulatory construct isn't fully created. I think the first companies that bring these tools through regulatory approval, what they're going to have to do, it's probably going to be some telehealth provider who's well funded. And what they'll have to do is bite off indication by indication. So they'll have to get approval for this chief complaint and that chief complaint and that chief complaint, and couple enough of these together where then maybe they can get approval for a general use case in a primary care setting, which would be a longer way off.
It's hard, but I still have to ask. I mean, so if the final approval is always still a human physician, why do you need any FDA approval? Because there's really conceptually not much difference between that physician doing a lot of Google research and just doing a lot of internet research themselves and coming up with an explanation, or having their assistant do it. Let's say another human,
but
a resident do the same thing, and then they check the work to make sure it makes sense.
Right.
And
So in in the circumstance where the physician is the final, touch point, that wouldn't be FDA approved.
And so is that currently how it is? Are we using
that's
these,
clinical
these
decision
tools
support
right now?
software. So
Okay,
that would be
that's okay right now.
That's okay. So that's carved out. FDA does regulate some clinical decision support software, but typically isn't regulating these tools. As long as the tool itself ultimately defaults to the physician. So what it's doing is, interviewing the patient, coming up with a presumptive diagnosis.
I understand now.
But that's not the real value, right? You laid it out at the outset.
No, yeah, eventually. No, if you're on a spaceship, maybe it's the Enterprise or something like that, and you need to have a holographic doctor on board because it doesn't take up any oxygen.
Yeah, but
It doesn't need food.
even diagnosing, flu or strep throat, something simple where you can couple a diagnostic test with an interaction with a large language model that's trained on that. I think that the tools now are capable enough to handle those lower risk interventions and know when to default to a provider, but nobody's developing the tools for those purposes because they're worried about, how they're going to be regulated and the cost of complying.
Okay, so maybe that's in these hearings, honestly, the conversations are not good because I don't think everyone understands AI, but I think you've laid it out very clearly for us on the questions we need to be asking, which is, you need to FDA, you need to start setting parameters for this future, so that the technology actually innovators have an incentive to develop the technology in the first place. Although if you're talking about assisting a doctor, I mean, why is the technology all that different? It's clearly not as advanced, but it shouldn't be that different, right? Because the goal for, even if it's clinician assisted technology, should be to provide a final answer. It just happens to have to need approval from a doctor. Is there something out there that's really impressive, by the way, as far as that kind of software is concerned?
Well, look, there's tools already that have been doing that layer on to electronic medical records and query physicians or look at lab results and make sort of a presumptive analysis or diagnosis and then queries the physicians. These kinds of tools, these sort of machine learning tools have existed for a while. And if you think about tools that radiologists are using to diagnose findings on x-rays, or pathologists are using to look at the slides, those aren't the large language models that we think about with like ChatGPT. Those are forms of machine learning where they're basically engaging in forms of pattern recognition. They're trained on closed data sets and taught how to identify certain findings by being shown a lot of those findings. And being
I wouldn't call it AI. Yeah, I would call it.
They are a form of AI.
Software.
It's machine learning is a form of AI.
Okay.
real opportunities that I think you're talking about is the large language models. So I'm doing an analysis right now where we're feeding the medical licensing exam to the different large language models to see which is the best doctor. But I think right now, based on my kind of cursory experience with these and doing some of this work, it looks like Claude from Anthropic may be the best at answering medical questions, better than Google's Gemini Advanced and better than ChatGPT 4.0.
Yeah. Well, it'd be interesting to see.
I'm not
where we
recommending
get
people
to.
go out and use it as a doctor, but it does
Yeah.
look to be the most reliable right now.
Yep. Dr. Scott Gottlieb, everyone says you don't need to see a doctor. Just Google it. It'll be fine.
That's not what I said.
No, that would be interesting. We look forward to continue to work with you on ways we can improve FDA policy because and I think in the end get people the access to innovations in healthcare that they need. So, Dr. Scott Gottlieb, thanks for being on again. Appreciate you.
Thanks a lot.